Good afternoon, students. Hope you are doing well and you are in good health. So today we're going to discuss about the virus, viroid, prion, and mycoplasma. So start with the virus, which is the most interesting topic nowadays. So before going to the details of the virus structure and all, so let's talk about a bit of history of the virus and virology. So, the scientific study of the viruses and infectious agents begins in the closing year of 19th century. Although Louis Pasteur and Edward Jenner developed the first vaccine against the viral infections, but they did not know about that the virus actually existed. So in the coming year, the first evidence of virus come from experiments with filters. I repeat again, experiments with filters that had pores small enough to retain bacteria. So in 1892, Dmitry Ivanovsky used one such filter to show that sap from a diseased tobacco plant remained infectious to healthy tobacco plant despite having been filtered. So finally, in the year 1898, one scientist, Martinus Bezzernick, called that filtered and infectious substance a virus, and this discovery is considered to be the beginning of the virology era. And finally, in the early 20th century, an English bacteriologist discovered bacteriophage. The bacteriologist named Frederick Todt. Okay. Now, what is actually a virus? So virus is actually comes from a Latin word which means venom or poison. So it can be defined, it is actually a submicroscopic entity consisting of a single nucleic acid, either RNA or DNA, which is surrounded by a protein coat which is known as the capsid and capable of replication only within the living cells of bacteria, animals or plants. So here the submicroscopic entity means the virus is actually very small to be seen by any compound or light microscopes. So that is why they are known as an ultra microscopic entity which can be seen only with the help of an electron microscope. That is why it's submicroscopic particle or agents. Okay, and single nucleic acid means it has only RNA or DNA but not both. Okay, and that particular nucleic acid is surrounded by a protein coat, and that protein coat is known as the capsid. Okay, and it is capable of replication within the living cells. So the virus actually can replicate only within the living cell but not outside of that particular cell. Okay, so sometimes that virus can be known as an obligate intracellular parasite. So here lies the question: why that virus is known as an obligate intracellular parasite? Now the answer. So why it is known as obligate cellular parasite? Because that viruses can multiplicate means here the replication, that means it can replicate its nucleic acid only within the host cells. So actually the virus utilize the cellular machineries for replication. That means it cannot divide by itself. Actually the virus don't have any kind of enzymes or some machineries to replicate its own DNA. That is why it will infect the host cells. Actually it hijack the host cell and use or utilize its machineries to synthesize several proteins, its own membrane, several amino acids, carbohydrates and lipids, and finally it can generate its an energy. So that is why the viruses can only survive only within the host cell. Okay, so that is why the viruses are known as obligate cellular parasite. Obligate means to restrict its surviving capacity only within certain organisms or host. That is why it is known as obligate cellular parasite. Okay, now this is the structural comparison of the viruses. You can see here that is the red blood cell or RBC, which is about 10,000 nanometer in diameter. Okay, compared to that red blood cell, you can see here that E. coli bacteria, which is about 1000 and into 3000 nanometer in diameter or its volume. Compared to that particular bacterium, you can see here that there are so much, there are large plethora of viruses is there. So you can see here that the smallpox virus is quite small. It is about 200 nanometer and 300 nanometer. Even in the bacteriophage, it is about 50 nanometer diameter. In case of the polio virus, it is about 30 nanometer. In case of the bacteriophage MS2, it is about 24 nanometer. And in case of the tobacco mosaic virus, it's quite small. You can see here that only the 15 nanometer in diameter. So the viruses are actually very small to be observed by some normal microscopes, even in the compound microscopes. So the virus can only be seen with the help of some electron microscopes. Okay, so this is the size comparison of the viruses. Okay, now go to the next slide. Yeah, that is a very much important questions when you are to learn about the virus. That is, are viruses living or non-living? This is the question. So actually the viruses are both or neither. That means it is at one side it is either living or non-living, and other side it is neither. That means it is not living or totally dead. So actually the viruses are living only within the host cell, and when it is outside the host, it is totally non-living or in a dead condition. Okay, so they have some properties of life but do not others properties. For example, the viruses can be killed, even sometimes crystallized like table salt. However, they can't maintain a constant internal state or homeostasis, which the homeostasis is a very important criteria present in living cells. That means the living cells can regulate its own physiological or anatomical, or you can say biochemical properties, but the viruses cannot maintain that kind of properties or internal state. So that is why the viruses are known as non-living entity or non-living agent. Okay, now go to the next slide. The living characteristics of virus and the non-living characteristics of virus. So the living characteristic is that the viruses contain the nucleic acid, that means the RNA or DNA, and it can reproduce or replicate, or you can say the multiplicate within the host cell or within the living cell, but outside the cell it cannot. Sometimes they can mutate. So this mutational property and the reproductive capability of the virus is a living characteristics. And on the side, the non-living characteristics of virus are it is totally acellular. That means they do not have any kind of cytoplasm or cellular organelles. So by the definition of a cell, either it is prokaryote or eukaryote, we know that the cell obviously have some kind of cytoplasm and some cellular organelles, but the virus is totally acellular. That means it do not have any kind of cytoplasm. Second one is that it carry out no metabolism on their own and must replicate using the host cell's metabolic machinery. I have already mentioned that particular point in my in last slide, that it cannot metabolize by itself. So it will hijack the host cell for its metabolic purposes. Okay, so in other words, viruses don't grow and divide. Instead, new viral components are synthesized and assembled within the infected host cell. So basically, the virus never ever divide or grow. Rather, instead of that, the viral components are synthesized within the host cell and assembled accordingly to form a newly formed virus within the host cell, and finally it get exit from the host cell after infection of the host cell. So this is the second point where the viruses can be defined as a non-living entity. And finally, the viruses possess either DNA or RNA, but never both. In contrastingly, in a cellular component or in a cell, has both the nucleic acids, DNA and RNA, because in the cell, to produce some proteins or enzymes, the DNA has to be transcribed to RNA, and finally the RNA has to be translated to form different types of proteins or enzymes, and that enzymes are important for you can say for the normal growing or normal metabolism of the cell. But as the virus is totally acellular, that is why it have only DNA or RNA but never both. Okay, now go to the next slide. So what is the difference between virus and bacteria? This is the important question, that bacteria is unicellular or one cell. It is made up of one cell. That means bacteria is made up of a cell. But in case of viruses, it is totally acellular. That means it is not made up of a particular cell. Okay. Second one, bacteria is quite larger than the virus. It is about 1000 nanometer in diameter in case of bacteria, but in case of viruses, it is quite small. I have already mentioned. The Latin name of the bacteria is Stix. Stix means it can stick with some host cell, but in case of viruses, the Latin name means it is poisonous, or the Latin name means it's poison. Okay. Finally, the bacteria usually treated with antibiotics, but the viruses are totally indifferent of antibiotics. That means the viruses cannot be killed or cannot be inhibited its action with the help of antibiotics. So the viral disease cannot ever be treated or cannot be cured with antibiotic chemicals. Okay, so these are the difference between bacteria and virus. So now, what are the characteristics of virus? I have already mentioned that virus is ultramicroscopic and cannot be observed using the light microscope. So the viruses can only be seen with the help of electron microscopes. Now they don't have any internal cellular structure. That means they don't have any cellular structure, cellular organelles, nucleus, cytoplasm, nothing. So virus is totally acellular. Next, the basic structure consists of a protein shell, which is known as the capsid, and surrounding the nucleic acid core. I will elaborate this point in next slide. Next point is that they contain either DNA or RNA, but not the both. Next one, they are incapable of metabolism. I have already mentioned why they are incapable of metabolism, and that is why they hijack the host machineries, host cellular machineries. Okay. Next one, viruses are smaller than the bacteria. They are active and replicate only within the host living cells. Next one, viruses lack cellular organelles like mitochondria, ribosomes, chloroplast, nucleus, everything. They don't have any kind of cell organelles. Next ones, the virus lack the enzymes for metabolic processes and protein synthesis. Finally, the molecules of viral surface impart high specificity for attachment to the host cell. I will explain that point. So now just briefly I will describe it, that virus surface contains several molecules, and that several molecules present in the viral surface acts as a, you can say, ligand, which will bind with the host cell, and finally with that with the help of that attachment, viral can infect the host cell. So, and that's attachment, that the attachment is very much specific for each virus and host cell. So a particular virus contain a particular specific molecule or ligand can only infect the host cell which contain a specific receptor for that particular virus, and that is why the virus surface has high specificity with for the attachment to the host cell. Okay, go to the next point or next slide. Now this is the very basic structure of the virus. You can see here that the virus is basically made up of a nucleic acid, it is either DNA or RNA, surrounding a protein coat which is known as the capsid. Nucleic acid and capsid both aggregately known as nucleocapsid. Sometimes some viruses have another envelope or membranous structure surrounding the nucleocapsid. This is known as the envelope protein or enveloped virus, and that envelope protein or envelope contain several ligand or marker molecules which is known as spikes or membrane proteins. So this made up the viral envelope. So the nucleic acid plus the capsid shell of a virus particle is often called the nucleocapsid. That means this one, the nucleic acid and capsid both aggregately form a nucleocapsid. Now what is virion? So virion is basically the complete virus particle. So that means the nucleic acid protein coat which may be surrounded by an envelope and which is very much infectious. So this is the virion, which is a complete viral particle which is quite infectious, is known as virion. Okay. Now the structure of virus. The size I have already mentioned, it is between 20 to 300 nanometer in diameter. The shapes either rod-like or spherical. The genomic material is either RNA or DNA, but never both. Importantly, the DNA or RNA either single-stranded or double-stranded. Okay. Now the nature of virus. So virus contain the nucleic acid, RNA or DNA genome inside a protein shell or capsid. This is a protein shell or capsid. This is the core, this is the capsid, this is the core or hollow lumen where the nucleic acid is present, and these are the spikes which is emerged from the capsid protein. Sometimes the capsid may be surrounded by a lipid bilayer membrane or envelope. So this is the envelope, okay, which contains several membrane proteins or which is known as the spike. The resulting complex virus particle is called the virion. That means this is a virion. Sometimes the nucleic acid is not surrounded by a capsid, rather the envelope is present. Okay. So basically the virus have three basic types, either naked or capsid virus, enveloped virus where the nucleic acid is surrounded by the envelope directly, or enveloped virus where the nucleic acid is surrounded by the at first capsid, and then the nucleocapsid is surrounded by a protein coat or envelope. Okay. Now go to the next slide. Viral structure, the capsid structure. Okay. So capsid is basically a protein coat which encloses and protects the nucleic acid of a virus. Okay, and it is the most mass component of the viral structure, and basically this is the capsid. You can see here, this is the capsid or protein coat where the nucleic acid is present inside of that capsid core, and capsid is actually made up of small subunits or smaller subunits. Each subunit is known as capsomere. Okay, so sometimes questions comes in your exam that what is capsid or what is capsomere. So capsid is basically a protein coat that encloses and protects the nucleic acid of a virus, and capsomere is actually the smaller subunit of the capsid. Okay, so finally the capsid together with the nucleic acid called the nucleocapsid. Now the characteristics and function of the capsid. Why do a virus have a capsid? Okay, so basically the capsid is made up of protein which is encoded by a viral genome, and capsid proteins are core proteins. So capsid shapes serve as the basis for morphological distinction. That means the shape of the capsid is actually used for the morphological morphological distinction of the virus. That means some viruses have different types of capsid, and based on the capsid protein or capsid structure, the viruses can be categorized morphologically or taxonomically. Okay, so the capsid protein is a basis of morphological characterization of viruses. Next one, the capsid actually protects the genome of the virus from atmospheric pressures. For example, UV light, some nucleases, some physical forces. Okay, otherwise it leaked or secreted by the cell. Okay, next one, the virus attachment protein interacts with cellular receptor to initiate infection. That means the capsid protein have some markers or some proteins, or you can say some ligand like structure, which help to interact with the host cell, and finally after interaction it will form an attachment and infect the host cell. Okay, finally the viruses deliver the genome in infectious form, maybe simply dump the genome into the cytoplasm or serve as a core for replication, some retroviruses and rotaviruses. Okay, now the virus classification, how a virus looks like. So based on the shape, it is basically three types, either polyhedral, helical or complex, and based on the envelope it is naked or enveloped virus. Okay, now go one by one. Now this is the isohedral or icosahedral capsid or virus, but the optimum way of forming a closed shell from identical subunits. You can see here these are the identical subunits which close and closed and form a core of a virus, and within the core the nucleic acid is present. Okay, so the minimum number of identical capsomere required is 12. So these are the capsomeres. Okay, these are capsomeres. This is one capsomere, this is another capsomere, this is another capsomere, and each composed of 5 identical subunits. So we can see here each capsomere, this is capsomere, this is one capsomere, it is composed of 5 identical subunits, or 1, 2, 3, 4, 5. So each capsomere is made up of 5 identical subunits. Each subunit is known as protomers. I repeat again, each subunit is known as protomers. Okay, PROTOMERS. Okay, so this is an example of a icosahedral capsid or icosahedral virus. This is a poliovirus. So another examples are adenovirus and herpes virus. Okay, now this is the helical virus. You can see here this is the helical virus, which is composed of a single type of capsomere stacked around a central axis to form a helical structure, which may have a central cavity, and within that cavity the nucleic acid is actually present, either RNA or DNA. So this arrangement result in a rod shaped filamentous virion. So you can see here this is the helical viruses where the helixes or helices form a filamentous or filament-like structure. Okay, so the nucleic acid is actually present inside the hollow cylindrical capsid. So this is a helical virus, you can see here, and these are the each subunits or capsomeres are within the helical capsid or helical structure. A lumen is formed, and within the lumen or hollow lumen the nucleic acids, either RNA or DNA, is present. Okay, some examples are rabies virus, tobacco mosaic virus. So this is a picture of tobacco mosaic virus. You can see here all the viruses. This is the viruses 1, 2, 3, 4. These are all different all viruses present, and all are in filamentous shape. Now the complex capsid or complex virus. So complex virus is either helical, or actually it is not purely helical or icosahedral. Rather it is composed of different other subunits which form a ultimate viral structure or virion, which is composed of sometimes a protein tails. You can see here these are the tails, or a complex outer wall. This is a complex outer wall. For example, poliovirus and bacteriophages. In case of polioviruses, it is a complex outer wall. You can see here this is a core wall, this is the inner membrane, and outer membrane is present, and within the outer membrane several spikes are there, and within the core there are DNA genome is present or RNA genome is present, and several virion enzymes are also present. Okay, and in case of bacteriophages, you can see here the tail sheath is present, a flexible tail sheath is or a spring like structure is present, a collar is present, some tail fibers are present, a base plate is present, some base pin or tail pins are present. So these are some accessory structure which is not present in helical or icosahedral virus. But complex capsid or complex virus contain several such accessory structures, and these accessory structures are adaptations for better infection within the host cell. Okay, now the viral structure, the enveloped and naked virus. Okay, so non-enveloped or naked viruses whose actually capsids are not covered by an envelope. You can see here this is a naked virus where the nucleic acid is, this is the nucleic acid which is covered by only a capsid protein, but not any enveloped protein is present. But in case of enveloped virus, the nucleocapsid, this is the nucleocapsid which is surrounded by a enveloped protein. In orange color you can see here this is the envelope protein, and envelope protein contains several spikes or membrane protein. Okay, so spikes are basically some carbohydrate protein complexes or glycoproteins that project from the envelope. For example, neuraminidase and hemagglutinin. These are two different glycoproteins which project or emerged out from the enveloped protein, okay, or enveloped virus, and can be used to attach to the host cell. Now little bit about the spike protein or neuraminidase or hemagglutinin. Okay, so these were the enveloped viruses present two major surface glycoproteins. Okay, glycoprotein means protein which have a carbohydrate moiety attached with some of the amino acids. Okay, amino acids means which is a building block or structural unit of a protein. Okay, so now these glycoproteins for hemagglutinin and neuraminidase, you can see here this is the neuraminidase molecule, the blue one, and the pyramid shaped or spike shaped hemagglutinin or in orange color. Okay, so these two glycoproteins actually recognize the sialic acid, which is you can say another glycoprotein or glycolipid present on the host plasma membrane, and have complementary activities. That means the these spike proteins make a complementary attachment with the sialic acid present in the plasma membrane of the host cell, and actually the HA or hemagglutinin binds with the sialic acid through its receptor binding site, and Na or neuraminidase actually receptor destroying enzyme, which is a receptor destroying enzyme that cleaves the sialic acid. Therefore, the functional hemagglutinin and neuraminidase balance is a critical factor for viral infection. Okay, and plays a major role in overcoming the host barrier and efficiency of sustained human to human transmission. Okay, so basically this is the neuraminidase and hemagglutinin. So neuraminidase cleaves the neuraminic acid present in the host cell and release the virus progeny from the infected cell. So with the help of neuraminidase enzyme, that particular virus will make an attachment with that of the sialic acid present in the host cell surface, and the virus get exit from the infected cell after release several virus progeny. Okay, and in case of hemagglutinin, which bind with the host cell surface, and in the host cell surface it again contains some sialic acid, and with the attachment of the sialic acid, the hemagglutinin make a complementary binding, and finally the viral particle will infect the host cell. Okay,